We will continue with the last part of the chapter of bacterial morphology. So we were learning about flagella. So flagella are long, thin helical appendages which help bacteria in locomotion. So flagella is basically organ of locomotion. It is a long helical filament. So based on the position and number of flagella, there are different types of bacteria. So it can be monotrichous when it has only one flagella at only one end. It can be amphitrichous when it has two flagella, one at each end. It can be lophotrichous when it has more than one flagella, all at the one end. And it can be peritrichous when it has flagella around entire body, all over the surface of the bacterial cell. So when bacterial cell does not contain any flagella, it is known as it is termed as atrichous. So we have discussed about the structure of flagella. The protein from which flagella is made up of, it is known as flagellin. And there are basically three parts of flagella: filament, hook, and basal body. So hook is the part that connects that long helical filament to the cell wall or to the bacterial cell body. And hook is that curved part. Basal body has two rings and one shaft or one rod. So gram-positive bacteria has only one ring that is embedded in the cell membrane, whereas gram-negative bacteria has two pairs of ring. One pair, which is known as outer ring, is embedded in the cell wall, and another pair, which is known as inner ring, it is embedded in the cell membrane. So as far as movement of the bacterial cell is concerned, the long helical filament rotates in a propeller-like motion, which causes rotation of outer ring and inner ring with respect to each other. So because of the rotation of those two rings, which requires energy, it consumes energy. And because of rotation of helical filament and those two rings, bacterial cell is moved forward. Okay, it is propelled forward. So the propeller-like motion or that helical motion of that filament is responsible for the locomotion of the bacteria. So this is the difference between gram-positive and gram-negative flagellar structure. Gram-positive cell has only one ring, which is embedded in the cell wall, and the rod goes to the peptidoglycan layer and further extends into hook and filament. Whereas in gram-negative cell, there are two pairs of rings. One pair is embedded in the cell membrane and another pair is embedded in the cell wall, which further extends into hook and then filament. Another external structure is axial filament. Axial filament is nothing but flagella. It is a type of flagella, but this special type of flagella extends all over the body of the bacteria. So it surrounds or it spirals around the body along the length of the bacterial cell. So this flagella is further covered by one membrane, which is known as outer sheath. So this flagella is present below the outer sheath, and that is why it is known as endoflagella. So axial filament is nothing but a type of flagella which is known as endoflagella. And this particular arrangement of flagella around the bacterial body gives the spirochetes that typical shape of corkscrew. So it is common in the species of spirochetes. Next appendages are pili, or singular is pilus or pilus. So pili are not as lengthy as flagella, and pili are not involved in any movement. So pili are not the organs of locomotion. These are tiny and hollow projections which extend from the bacterial cell surface. So the main role of pili is to help bacterial cells adhere to the surfaces or attach to the surfaces. So there are two types of pili. The first one is conjugation pili. It is also known as F pili or sex pili, and the other one is attachment pili. The other name for attachment pili is fimbriae. So conjugation pili is lengthier. It is more in length as compared to attachment pili. So conjugation pili, as it is known as sex pili, it helps in conjugation of two bacterial cells. But this type of conjugation does not lead to any kind of reproduction. This is not the way by which bacteria reproduce, or this type of conjugation will not lead to uh, uh, any birth of any new bacterial cell. So this type of conjugation is basically helps in genetic transfer from one bacterial cell to other. So DNA and genetic material is transferred from one cell to another with the help of conjugation pili. Another type is attachment pili or fimbriae. These are shorter in length as compared to conjugation pili, and they extend from the cell surface, and they basically help in adherence of the bacterial cell to any surfaces. For example, if you are keeping those bacteria in a nutrient broth in a test tube, so attachment pili or fimbriae will help those organisms to attach to the wall of the test tube. Or for example, few organisms are sensitive to presence of oxygen. They require a lot of concentration of oxygen for survival. So what will happen when you inoculate such organisms in nutrient broth in a test tube? They will try to accumulate towards the surface to acquire or to get more oxygen from air. So such organisms, such microorganisms will come together near the surface, and they will form a whitish layer, a whitish film just below the surface of the nutrient broth. So they get attached to the surface of the nutrient broth with the help of attachment pili or fimbriae. So attachment pili also contributes to the pathogenicity of the organism. Pathogenicity is ability of organism to cause disease in the host body. Now, the last part of external structure of bacterial cell is glycocalyx. Glycocalyx is basically a polysaccharide or gel type secretion which is secreted by bacterial cell on the surface. So not all bacteria possess the property of glycocalyx. Only few bacteria can secrete this sticky substance around them. So the main role of this sticky glycocalyx or polysaccharide substance is the protection of bacterial cell. So it is present in the form of a loose gel. So if the thickness of this glycocalyx layer is very much, or it is very thick in nature, it is known as capsule. And if it is present only as a thin layer around the cell, it is known as a slime layer. Okay, if it is very thick, then it is capsule. If it is only thin layer, single layer, then it is known as slime layer. It is slime layer is less tightly bound as compared to capsule. So for both these capsules and slime layer, the role is a protection of the cell. So because of these, the cell surface of the bacterial cell becomes very slimy in nature. So whenever they are causing diseases in the body of human beings, what will happen? Our body will trigger the immune response, and phagocytotic reaction will be initiated. So phagocytic cells will try to capture these invading organisms and destroy them. So what will happen? Because of this slimy surface, these microbial cells can easily evade the attack by phagocytic cells. So it becomes very difficult for phagocytic cells to capture such slimy organisms and destroy them. So that is why capsule and slime layer add to the virulence mechanism of the infecting organism. So the diagram that you can see in the on the slide is the example of slime layer, or it is in this case it is the picture of the plaque, dental plaque. So it is formed by the bacteria which infects oral cavity. They secrete this slime layer all over the teeth and gums, and that slime layer is so strong, it is so strongly attached to that surface that normal brushing cannot remove it. So you cannot remove it with the help of normal brushing, and you have to take a help of dentist to remove that slime layer. So that strongly it gets attached to the surfaces. That was about glycocalyx. After that,